Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your determination of extraneous matter in a potato chips. See, potato chips are made up of a potato and there is higher chances that it will be contaminated with the extraneous matter. Contaminated with the extraneous matter either during the harvesting of the potato, harvesting of the potato, or during the processing or storage of this potato chips. Processing or storage of this potato chips. Okay, there is highly chances that. Okay, and the introduction part of this extraneous definition of this extraneous matter are already uploaded in our account. You can go and see the video related to this, that is the determination, and the topic of the video is determination of the extraneous matter in a cottage cheese. Here what should we do? I'm going to tell you the basic of this extraneous matter. Extraneous matter are simply what any foreign matter or a material which are added into the food product. Okay, and this addition are either intentionally or either unintentionally, but ultimately it will impact the health of the human being, impact the health of the human being. So that's why it is important to determination of this extraneous matter. And why we are using the potato chips? Because this potato chips are consumed mainly by our children's. Okay, they are what? They are consumed mainly by our children's. And children's have a weak immune system. Okay, if someone have a weak immune system, like children, like older people, like a pregnant lady, then the foreign material attack their body very easily because their immune system are weak. That's why it is very important that we have to determine this extraneous matter present into a potato chips. Okay, now the example of the extreme matter, either they are flit, either they are insect, either they are the rodents. So anything which are not the ingredients of the food product, but they will add it into a food product or they will contaminate the food product. So this is called what? This is called extraneous matter. Now come to the next slide. Okay, now the next topic is the objective of this experiment. So here the objective of this experiment is simply we are using our different technologies or different techniques so that we can determine the extraneous matter which are present into our potato chips. Okay, so what we have a food sample, what we have a food sample here are potato chips. Potato chips are what? It is a food sample. So what should we do? We use our different technologies. So can we isolate this extraneous matter present into our potato chips? Now see what is written in the slide. The objective of this laboratory is to utilize the techniques to isolate the extraneous matter from a potato chips. Here the food sample are what? Here the food sample are the potato chips. Now the next one is your principle of this experiment. See, the principle are same that we already discussed in the previous lectures. So here I'm going to explain you. What should we do? First of all, we have to separate the extraneous matter present into a food product, and this separation are done either on the basis of the particle size, sedimentation, and the affinity for a oleophilic solution. Okay, and once it's separated or isolated, we examine this extraneous material with the help of microscope because it cannot be seen by our naked That's why we are using the microscope for the visualization of this extraneous material. So first of all, we have to separate this. For the separation, it based on a particle size, sedimentation, and affinity for a oleophilic solution. We uses the different technologies for their separation. Once it's separate, it will be trap into a filter paper. Then this filter paper should be examined under a microscope. Now see what is written in a slide. Extraneous material can be separated from a food product by a particle size, sedimentation, and affinity for a oleophilic solution. Sedimentation and affinity for a oleophilic solution. Once isolated extraneous material can be examined microscopically. Okay. Now the next one is the chemical used in this experiment. So here we are using the three chemical for performing this experiment. The first chemical is a 95% of ethanol. We require 95% of ethanol. Okay. Case number 6417-5, and the hazard related to this chemical are what? They are highly flammable. Okay, if it is highly flammable, so we have to avoid it using this ethanol near the flame. Okay, now the next chemical is heptane, and the volume of the heptane will be a 9 ml. Case number 142-82-5, and they are what? They are harmful, they are highly flammable and dangerous to the environment. Now the harmful means it is harmful for the human being and animals. Okay, so we have to avoid it to come in contact with this type of chemical. So when we are using this chemical, we have to wear a proper glove, so it will not will come in contact with our eyes. And also we have to avoid the avoid this vapor to breathe in. Okay, so this is the hazard related to the heptane. Now the next or the last chemical is your petroleum ether, and the amount of the volume of this petroleum ether is 200 ml. That is case number 803232-4. Okay. Now the hazard related to this chemical, that is your petroleum ether, is it is harmful, highly flammable, dangerous to the environment. If any chemical are dangerous to the environment, so when we are disposing that type of chemical, we have to dispose it into a waste, hazardous waste. Okay, so these are the list of the chemicals that we used in our experiment. Now the next one is your reagent. Okay, so ethanol 60%. So what should we do? 1 liter use 95% of ethanol to prepare 1 liter of 60%. What should we do? We take a ethanol and dilute it with 632 ml of 95% of ethanol with a water to a 1 liter. Okay, now the next one is your hazard precaution and waste disposal. We all know that we are using two chemicals that are fire hazards, like your petroleum ether, heptane, ethanol. These three are what? They are fire hazards. So we have to avoid it. These chemical, sorry, we are avoid using this chemical near the flame, and we also avoid the breathing of the this vapor, and we also avoid the contact that type of chemical with the skin. Okay, and along with it, we have also followed the normal laboratory procedure, like we have to wear a glasses, we have to wear a lab coat, we have to wear a proper shoes, and even we have to avoid to eating anything inside the lab. And this heptane and the petroleum ether are wasted that can be disposed under a hazardous waste, and the other waste should be drain out by using the water rinse, and along with it we have also followed the protocol which are given by our institution. Okay, now see what is written in the slide. Petroleum, ether, heptane and ethanol are fire hazard, so we avoid open flames, breathing vapors and contact with the skin. Otherwise, adhere to our normal laboratory safety procedures like wear a safety glasses at all time. And one more thing that I'm going to add here, wear a lab coat. Okay, wear a gloves and wear a proper shoes. Wear a proper shoes. So these are the safety equipments or that we have to wear. Okay, heptane and the petroleum ether wasted must be disposed as a hazardous waste. Other waste may be put down the drain using a water rinse. So these are the hazard precaution and waste disposable slide. Now the material used in this experiment, first of all, we required a beaker of 400 ml and we required a Bushnell funnel, filter paper, glass stirring rod, graduated cylinder of 1 liter so that we can measure the 95% of the ethanol, and we also required a ice water bath. So here we required the material, that is your beaker of 400 ml, Bushnell funnel, filter paper, glass stirring rod, graduated cylinder of 1 liter, and this graduated cylinder are used to measure our 95% of ethanol. And at the last we required a ice bath. The equipment that required for performing this experiment, the first one is your hot plate. Okay, so this requirement of the hot plate is what? For heating the material, for heating the materials. Okay, microscope are required for visualization of extraneous material. Okay, top loading balance are used for weighing the sample, weighing the sample. And at the end we required a water aspirator system, and it is required to carry the vacuum. Okay, so we required hot plate, microscope, top loading balance and water aspiratory system. Now the next one is your procedure. See, first of all we have to take a 400 ml of beaker. This is your 400 ml of beaker. Now in 400 ml beaker, we have to add a 25 gram of potato chips. Okay, then we have to crush, suppose that this is your potato chips. Now with the help of glass spatula or a glass stirring rod. Suppose that this is your glass stirring rod or spatula. Okay, with the help of this, what should we do? We crush it. Okay, we crush it into a small pieces. Then inside the hood, so that it will not catches the flame, we add petroleum ether into it. Now what should we do? We add petroleum ether, and when we added this petroleum ether, we cover it and leave it for a 5 minute. Okay, now after 5 minutes we decay this petroleum ether from the chips through the filter paper, and again after that we add a petroleum ether to the chips, and again we leave it for a 5 minute and decay through a filter paper, and let a petroleum ether evaporate from the chips in a hood. So first of all, in a hood we add petroleum ether and cover the chips. Okay, and leave it for a 5 minute. Then we decay the petroleum ether from the chip with the help of this filter paper, and again we add the petroleum ether into the chip and leave it for a 5 minutes and decay through the filter paper, and again we allow the petroleum ether to evaporate in our chips. Then after that we uh, transfer the chips into a 500 ml of trap flask. Now from the beaker, what should we do? We transfer it into a trap flask. And into it we add a 125 ml of 60% of ethanol. And how we prepare this 60% of ethanol, this video related to the topic of this, the video are already uploaded in our account, and the name of the lecture is solution preparation. You can go and see the video. And when we added 60% of ethanol, we boil it for a 30 minutes, and during boiling, we mark the initial level of ethanol on a flask. During boiling and at the end of the boiling, replace the ethanol lost by evaporation as a result of boiling. So this is about the procedure of this experiment. Then after that, we cool it into a ice water bath. Okay. After cooling, suppose that this is our trap flask, in which we add all the things and we evaporate the ethanol. Then we cool it under a water bath, and again we add a 9 ml of heptane and mix it properly and leave it for a 5 minutes. And again we add a 60% of ethanol to the flask so that only the heptane layer is above the rubber stopper. Okay. So in a excess amount of ethanol we are going to add into a flask so that only the heptane layer should come above the rubber stopper. And after that we allow the heptane layer to form at the top and trap off the heptane layer with the help of the filter paper, and it with the help of the we trap the heptane layer and filter it with the help of this filter paper in a Buchner funnel. Then we add a 9 ml of more heptanes into a solution and mix it properly, and again we stand until all the heptane layer are come to the top. Then trap off the heptane layer and filter it through a filter paper. That is new piece of the filter paper, not the piece used in a part third and fourth in a Buchner funnel. So here what should we do? We use a fresh filter paper. Now we examine the filter paper into a microscope so that if there any extraneous material are present, it should be visualized. So this is all about the determination of the extraneous material into a potato chips. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box. And for a further detail, you can follow the ANALYST Academy on different platforms like your Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ANALYST Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450. You can also visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe. If you have any query, just let us know in a comment box.